we're going to change the U just to get the same amount of how much cells you want to process per second. Cool. So I'm also going to show you why also this design is popular in a little bit. So I'm going to tell you a story about two guys who works in your field and also publish papers in your field, and to discover more about single cell heterogeneity using microfluidics. So let's start with Sergey here. So this guy was really interested about genomics, and then he worked at the same lab with Benjamin. And Benjamin was kind of a microfluidic guy. Sergey is kind of like a genomics guy. And they published a paper where they basically showed a way that they can do PCR in droplets. So you can have here two inlets, and that have different or multi-phase interaction, where you have water, oil. When you mix them together in such channels, you get droplets. They put DNA in these droplets, and then they put them in the thermal cycler. And they found that they can actually get a PCR signal. They can do PCR in these droplets. And then they published this paper in Analytical Chemistry, where you can see the same. Do you remember the channel design? 3 inlets. They mix them together, one with oil, one with water. And then basically they put also cells in them. And they can do single cell genomics. They can actually see each cell will be inside the droplet. They can lysis, and then they can, within these reagents, one of the inlets, they can have PCR reagents, and they can also do PCR with signal in it. Now, did any of these give you any hint of who's these two guys are? You can raise your hand, or you can answer.
I know.
Did they go?
Serge Saxonoff is involved with 10x, right? Yeah. He's part of 10x Genomics.
Yes, awesome. So they are now, so as you can see, the device redesign, they are now holding like $1 billion company, 10x Genomics, right? And now they are the head of this company, which does single cell sequencing. And we actually have this device in our lab, which is also going to be in every other lab, and they use microfluidics to solve this problem and to approach this. And their publications, so they only published the 3 papers, right? But the publication that mention their device is increasing over time. So now they have like maybe 700 publications that mention 10x Genomics, right? And now you can discover what you can see here, single cell heterogeneity between this. And this really facilitates us researchers to actually move on to the next step and discover more cell heterogeneity, right? And so you can see this is Nature Medicine, Nature Biotechnology. They're all using 10x Genomics to discover single cell heterogeneity. And also, they are now contributing to the COVID-19 analysis. So I just saw this, so I thought it would be fun for you guys to read. So really, microfluidics, what I want to say is that it really increases the throughput. It's really important. Now it's going to be in every essential lab, basic lab research, or even clinical, right? Even clinical research using it. So now